Hello students, welcome to AskPrep. My name is Sakshi and we are talking about the chapter Cell, class 11. In case of any doubts or question, please reach out to us through the comment section. And also, if you're looking for an online batch, then our upcoming batch is starting up soon with a limited seats. So reach out to us for that also. Okay, now before we start with the various organelles in the cell, we will first of all talk about the cytoplasm. So cytoplasm is the fluid that the cell enclosed by the plasma membrane or the cell membrane. So the liquid which is present inside, the fluid which is present inside the cell, and it is confined via the plasma membrane is the cytoplasm. It is less viscous. Viscous means viscosity क्या होती है? Viscosity is the thickness of a liquid. अब इसमें अगर if 80% of water is present, that means its viscosity is very less. So it is less viscous. It is also called as cytosol. कुछ books में इसका नाम cytoplasm का cytosol भी दिया गया है. Okay, so अब इसके अंदर present क्या होता है? It contains dissolved nutrients. Whatever the nutrients required by the cell organelles, it is present here. It also has the dissolved waste products. अब cell है, functioning है, excretion भी होगा, waste products भी होगा. So these are also present inside this cytoplasm and then get removed from the cell. Now, organelles are embedded in the cytoplasm. जो जितने cell organelles के बारे में अभी हम आगे बात करेंगे, they are all present inside the cytoplasm. To some extent, it also controls the shape of the cell. अब आप, you can very well imagine that there is a fluid which is present inside the cell, so that also gives a little shape to the cell as well. Okay, now the cytoplasmic streaming. Streaming से word जैसे ही आता है तो हमें क्या समझ आ रहा है कि something is flowing inside. So the movement of cytoplasm within the cell. अब cytoplasm ऐसे तो है नहीं है, it is a liquid. So it is not confined to one place only. It is just moving around. So what is the function of this? It is the transport of nutrients and Okay. So cytoplasmic streaming, its function is the transport of nutrients within the cell organelles, exchange of material. अगर एक जगह से दूसरी जगह कुछ material जाना है तो वो कैसे जाएगा? It is within due to this the presence of cytoplasmic streaming. Now the factors on which this streaming, this flow depends is the temperature, pH, viscosity, hormones, and light. Under these various conditions, external conditions, the cytoplasmic streaming gets affected. More तेज हो रहा है, कम हो रहा है, टेम्परेचर ज्यादा है, उसका इफेक्ट है. pH जिसमें सेल पड़ा है, उसका इफेक्ट आ जाता है. हॉर्मोन्स जो रिलीज हो रहे हैं, and also to the light as well. Okay, now we talk about the endoplasmic reticulum. Endo means something which is inside. Plasmic is the word coming from plasma, and reticulum means the network. So from the name, as we can see, it is the network of some tubules which are present inside the plasma. Which plasma are we talking about here? The cytoplasm. So it is a complex network of membrane-bound tubules. So tubules है इस type के. ये देखिए जैसे ये डायग्राम में दिए गए हैं. So these can be rounded, oblongated, or tubular-like, and it is different in different type of cells, depending on their functions. कि क्या क्या function perform कर रहे हैं. Like in the lower organism, it is not very well defined. But in the higher organism, it is well defined and has and performs the specific functions. Okay, now about the types. Rough endoplasmic reticulum and the smooth endoplasmic reticulum. So RER and SER. So RER ऐसे जैसे हमें नाम से ही पता लग रहा है कि R rough है, कुछ roughness है उसके surface पे, that we can see in the appearance. And smooth, that means the surface is very, very smooth. Why is it so? We'll talk about now. So first of all, rough endoplasmic reticulum. Now, if you can just see in the picture right over here, we can see some tiny granulated structures which are present on these oblongated tubes. So these are the ribosomes. So these ribosomes, the presence of these ribosomes make it appear rough, right? So that is why it's got this name RER. Now, one more thing, it is always attached very close to the nucleus. Nucleus के बहुत पास होगा. Or ribosomes, as we know, is the site of protein synthesis. So protein manufacture hota hai ribosome mein, and then it get transported to the other parts via the endoplasmic reticulum. Now, this is the RER, and this one is the SER, the smooth endoplasmic reticulum. You can see it. It is a continuous network. पहले ये रफ एंडोप्लाज्मिक रेटिकुलम है, उसके बाद ये स्मूथ भी उसके साथ कंटिन्यूटी में ही है. देयर इज नो गैप बिटवीन दैम. Now, smooth, as the name suggests, the surface is smooth. Why? Because they lack the ribosomes. There are no ribosomes present here in SER. So then what function do they play? They play the function of fat molecules and lipids manufacturing, right? So lipids and fats, they are manufactured here, and it also help in detoxification of chemicals that are into safer water soluble substances. So ये SER का main काम है to manufacture proteins and fat and lipid, while ribosomes का काम है manufacture synthesis of protein which are present in RER, and in SER they are lipids and fats. Okay, so that is the main difference between these two. Now, the significance. What is the role of this endoplasmic reticulum? First of all, we have studied about the protein and lipid manufacture. Protein, हमने अभी बात की, is manufactured in which? Yes, it is. It manufactures in RER and lipids in SER, right? So, and building up of the plasma membrane. So, कुछ हद तक ये help in building up the plasma membrane also secretes enzymes and hormones. Now, ER acts as a channel of transport material to different part of the cell. अब यहाँ पर सिंथेसिस हो रहा है प्रोटीन्स का, इधर लिपिड्स का हो रहा है, फैट्स का हो रहा है. तो इन ट्यूब्यूल्स का क्या काम है? टू ट्रांसपोर्ट दिस मटेरियल टू रेस्ट ऑफ दी ऑर्गेनेलीज. अब सबको कुछ इस सामान की जरूरत तो होगी, सो यहाँ पर ट्रांसपोर्टेशन का काम किया जा रहा है, राइट? डिटॉक्सिफाइंग पॉइजन एंड ड्रग्स मीन्स कि नॉट द एंटायर, लाइक को ज्यादा पॉइजन दे दिया उसके आगे तो सेल सर्वाइव नहीं कर पाएगा. इट विल बी वेरी डिफिकल्ट फॉर द सेल टू सर्वाइव. So, but the poison and harmful chemicals in some extent are controlled by this SER because it helps in the detoxification. Now we come to the Golgi apparatus. और कहीं कहीं इसको Golgi bodies भी बोला जाता है because it is again a network of membrane bound vesicles. Right? They are arranged in parallel to each other in like a sac. Like we can see this. ये देखिए, ये they are all parallel to each other. एक दूसरे के बिल्कुल parallel चल रहे हैं. Called cisterns. Now ये इन phases को बोला गया है cisterns. Now इसके two phases होते हैं. Two phases in the sense two sides होते हैं. One is the cis phase and other is the trans phase. So cis phase कहाँ है? अब जैसे हमने प्रीवियस उसमें बात की थी एंडोप्लाज्मिक रेटिकुलम में, दैट इट इज आल्सो कनेक्टेड टू, आई विल गो बैक टू द प्रीवियस स्लाइड, जस्ट लुक एट दिस, इट इज आल्सो वेरी वेरी क्लोजर टू द न्यूक्लियस. सो वन एंड इज अटैचिंग टू द न्यूक्लियर मेम्ब्रेन RER का, and the other is to SER. Likewise, SER का one end is attached to RER and the other end over here will be the Golgi apparatus as we can see it over here. Right? यहाँ पर diagram में clearly दिखा जा रहा है, right? So this is the cis phase and the trans phase. Now, cis phase is located near the nucleus in the animal cell. ये है वो cis phase जो nucleus को face कर रहा है, उसकी तरफ facing towards the nucleus. And the freely, and sometimes in the plant cell we will see a little difference here, that it is distributed freely, and the other name which was given to it is dictyosomes. Now, अब ये नहीं है, Golgi apparatus is not present in all the kind of cells. It is present in some, and there are a few exceptions where these cells it lacks. So sperm cells of bryophyte, pteridophytes, sperm cells of animals, RBC of animals, cells of some fungi and prokaryotes. So these cells lack Golgi bodies. Now, the structure. सबसे पहले इनका structure कैसा है? We will talk about that. So there are three main components: cisternae, cisphase and transphase, जिसमें आएगा. Then there are tubules and the vesicles. So cisternae are the fluid-filled sac-like structure. They are stacked in piles. ये देखिए जैसे हमने यहाँ पे किया. These are not empty. They are the fluid-filled structure. They are arranged in the concentric fashion. Now concentric का मतलब कि एक यहाँ पर डॉट आप अगर इमेजिन करें सेंटर में and उसके around, right along with the around that, keeping something in the center and uske around jab kuch घूम raha hai, so that shape or the fashion is called the concentric fashion. So isme bhi keeping this in mind, uske this cis face is facing the as we have discussed, the smooth endoplasmic reticulum, and then the rest is arranged in the concentric fashion. So it is the cis phase is also called the forming fashion. The young part of the cell, the young part of the organelles are here in the cis phase, and the older, the maturing ones they come over here in the trans phase. Now, tubules. The network of tube-like structure arise from the peripheral area of cistern here, sides में से. So tubule-like structure, they are present over here. The vesicles, membrane-bound organelles, which contains, now vesicles need not be attached to the main bodies here. As you can see it in the diagram, they are the dense granules which are there, which are present in the cytoplasm, free from, they're not attached to the cisternes. So they are just lying undetached and they appear just lying in the cytoplasm. About the significance, इसका काम क्या है Golgi apparatus का? Material manufactured in ER is dispatched to various parts inside or outside the cell through Golgi apparatus. अब imagine कीजिए कि आपने कुछ, you have ordered something online, right? So you've chosen a site and you placed an order there. Now from that side they will inform their warehouse, so to pack it and then send it across. So this can be compared to a warehouse where the manufacturing was done. किसकी manufacturing पढ़ी है अभी हमने ER में? We have studied about the proteins and the lipids, right? So proteins and the lipid, whatever is manufactured over there by the ribosomes, so it is sent over here in the Golgi apparatus and यहाँ से उसकी packaging होके and then it goes to the various parts, right? Storage of various cellular secretion and synthesis of lysosomes. So lysosomes की synthesis भी यहीं होती है in the Golgi apparatus. So now we know the major significance of Golgi apparatus. Its work is to dispatch the various secretions, the various manufactured material, whatever ER has delivered to the Golgi apparatus, और यहाँ से through these various tubules it reaches to the different parts of the cell. Now about the lysosome. Lysosome के बारे में हमने अभी पढ़ा कि they get synthesized by the Golgi apparatus also. Now, membrane-bound vesicles containing variety of digestive enzymes. अब यहाँ पर, this is the picture here, इसके अंदर बहुत सारे digestive enzymes present हैं. So, these are a few names of the digestive enzymes that are present here. Proteases, as the name suggests, it helps in digestion of protein. Lipases, lipids ka, nucleases, nucleic acid, glycosidase, and phosphatase. So they play different roles according to the requirement. Now, the significance. So digest waste materials also, defense against bacterial and viral infection. अब ये जो juices secrete करता है, उससे in case of any attack of a microbial infection in the cell, in the body, so this works against that. This also acts as a defense mechanism of the cell. Now during the cell damage कई बार क्या होता है कि lysosomes जो हैं ये ऐसे तो सेल के अंदर प्रेजेंट हैं, राइट? सो दे बर्स्ट आउट एंड द एंजाइम डाइजेस्ट इट्स ओन सेल। अब वो कैसे होता है कि पहले तो सबसे पहले जो इनकी मेम्ब्रेन है, एज यू कैन सी इट ओवर हियर, दिस मेम्ब्रेन इज वेरी थिक, बट समटाइम्स व्हाट हैपेंस कि कुछ रप्चर हो गया, द सेल फंक्शन, द मेल फंक्शन हो गया सेल में, then the juice gets released out from the lysosome, and then these lysosomes start digesting its own membrane and spread it out in the cytoplasm and start digesting whatever it gets. So in those cases it can digest its own organelles as well. That is why it is also called as the suicide bag of the cell. Suicidal bags क्यों कहा गया है इनको? क्योंकि in case of in some cases, whenever the cell has some malfunctioning or not damaged or not functioning in the proper manner, then what happens? These lysosomes, they end up digesting their own cell organelles and hence rupturing the cell and causing a great damage to the cell. So that is why it is called as the suicidal bag. So after lysosome, we'll talk about the vacuole. So vacuole is a fluid-filled membrane-bound sac. Sac क्या होता है? Sac is a big bag, right? So this, as you can see it in the picture, so this is a big bag which is filled with the fluid. Now, what all is there in this fluid? It has water, and sap, minerals, sugar, amino acid, proteins. It also helps in carrying the toxic and waste secretions. Okay. Now, is it present in the animal cells or not? Now, some animal cells do show, but a few do not even, right? They do not have the vacuole in them. So it is not present in all the animal cells. Okay. So, but in plant cell, yes, because the plants need to store a lot of food inside, a lot of water and the nutrition that is required by the plant. It has to be stored inside. So, plant cell has significantly big vacuole in them, right? So the tonoplast is present. Tonoplast is a membrane around the vacuole. The membrane covering the vacuole, and it is selectively permeable. What do we mean by selectively permeable? Means doesn't allow everything to go inside. Only some things are to pass through it, not everything. So that way it is very choosy. It is very selective by checking out the material, whatever is going in and out of the this vacuole, right? So the plant cell has sap and provides a rigidity to the cell because plants के पास अपना कोई skeletal system नहीं होता है. So we will see it more often in many parts, many different cell organelles that provide the rigidity to the plant because that is what they need more, right? And it also, as we have just discussed, it is also a storage for amino acids, sugars, and proteins because उनके पास storage के लिए कोई specific organ नहीं है. That is why the vacuoles are present and they're significantly big in size in the plant cell, right? In some animal cells, like amoeba and paramecium, the different type of vacuoles are present, like the digestive vacuoles, as the name suggests. So they help in the digestion of the food. And the contractile vacuole. Contractile vacuole's role is to excrete the stuff outside the cell, whatever the material that is not required by the cell, it expels it out of the body. So that is done by the contractile vacuole. So I hope whatever we have done so far is very much clear to you. In case of any doubts, please reach out to us. Okay, so now we come to the next organelle, that is the mitochondria. So mito means thread and chondria means the granules, right? So these are rod-shaped organelles. Rod-shaped इनको क्यों कहा गया? Because they are very minute and microscopic. So that is why they are called rod-shaped. Bounded by a double membrane. Double membrane, as you can see it clearly in the diagram, the orange one is the outer membrane and the pink one is the inner membrane, as it is mentioned over here also. And there is a space in between, which is called intermembrane space, right? Now, this is the site of respiration. So respiration takes place here and Respiration is a very, very important part of all the cells, as because of that, the ATP is also released, the energy also released, and without energy, none of the cell can perform by their own functions, right? So that is why it is also called as the powerhouse of the cell. Now, about the significance, क्या role है mitochondria का? We have studied already, we have talked about it, that it is also called as the powerhouse of the cell. क्यों? क्योंकि यहाँ पर ATP generation होता है, respiration takes place here, energy is released here. ATP क्या होता है? Adenosine triphosphate, right? It can synthesize their own proteins and they have their own DNA and ribosomes also. अच्छा, इसके पास अपना डीएनए प्रेजेंट होता है, अपने राइबोसोम्स होते हैं. वाई? बिकॉज यहाँ पर बहुत सारा काम हो रहा होता है. अ लॉट ऑफ work is to be done so that the respiration, the breaking down of the food, of the material is has to be taken place, and the release of ATP has to be done. So that is why it has its own ribosomes for the protein synthesis. It has its own DNA. Now we'll talk about the structure of mitochondria. Now here we can see the outer membrane is the orange one that is reflected here in the diagram, and the inner one is the pink color, right? So there is a gap also between these two layers. This is called the intermembrane space, right? Which is mentioned over here also, right? It is continuous. This outer membrane is continuous throughout, right? When we talk about the inner membrane, it is deeply folded. Deeply folded means there are multiple folds inside this. Why are there multiple folds? It is to increase the surface area. The more and more the surface area will be, the more it is convenient for the ATP generation, right? So it gets a larger surface area. Like if you imagine आपके पास अगर इतना area है and you have to place a wire in between that. So if you place it like this in a straight manner, then it will require one space. Or if you curl it, put it in the curly manner, then it will require more area, more surface area within the specific given portion to you, right? Likewise, the inner portion, the inner membrane is folded in multiple manners so that it gives the larger surface area. Now, these folds, these are called the cristae, right? As we can see it here in the diagram very clearly. Now, do not confuse cristae with cisternae, which we have studied in the Golgi apparatus also, right? So there are many compartments which are present. These are called the cristae. And the matrix is the ground substance which is present and it is enclosed by the inner membrane. Okay, I hope this much is clear to you. Please do let us know in the comment section. And if you want an online batch also, do reach out to us. Thank you. So after mitochondria, we come to the plastid. So plastid is the organelle which is specifically present in the plant cell. There are certain pigments present in the plant's plastid that play various role according to their function. The three type of pigments are chloroplast, chromoplast, and leucoplast. Now, first of all, we'll talk about the leucoplast. These are the colorless pigments. अब ये present होते हैं inside the plastids and इनका role है is to storage of the food. As we have discussed earlier also, कि in plants there are various organelles or the various things we will study about that helps in the storage, right? So यहाँ भी these leucoplasts, they help in the storage of starch, oil, and proteins, right? Chromoplast, chromo का मतलब यहाँ पर है color. The word chromo comes from the word color, and plast is the pigments, right? So these are the color pigments, जिससे कि fruit को, vegetable को, और flower को एक specific color मिलता है. That is because of these chromoplasts, right? So they are responsible for imparting the colors, right? Chloroplast, we are very much familiar with this. What is a chloroplast? it contains a green pigment called the chlorophyll. We have spoken about it in our junior classes also. Chlorophyll का क्या काम है? It helps in the photosynthesis, right? And it also imparts the green color to the leaves. Leaves के अंदर जो green color होता है, that is because of this pigment, that is the chlorophyll, right? It helps in photosynthesis and also it is called the kitchen of the cell. जैसे leaf को बोलते हैं, it is the kitchen of the plant, right? Likewise, this chloroplast is the kitchen of the cell. Why? Because यहाँ पर chlorophyll present है, जो help करता है photosynthesis में, right? Talking about the structure. अब इसका structure कैसा होता है of the plastids? Now, it is a double membrane structure, as we can see it here in the diagram. There are two membranes present. The stroma contains the matrix inside, this one, the homogeneous proteinaceous fluid, which is present inside here. It contains DNA, ribosomes, and the various enzymes. Now, thylakoids are present in stroma. ये जो stack आप देख रहे हैं, ring का stack, जैसे coins को अगर one above the other रख दें, तो it forms the stack of a coin, right? So likewise, these thylakoids, they are present in the stroma, and they are present in the stack formation, right? Are arranged in a stack, it is called, which contain the chlorophyll. So मतलब chlorophyll जो है इसके अंदर present होता है, right? In stacks के बीच में. These stacks of thylakoids, they are called grana. So एक stack जिसमें various thylakoids present हैं, उसको बोलेंगे grana, right? Now there, if you can see it, observe it very, very carefully here in the diagram. They are the flat tubules that connects granal thylakoids. मतलब ये दो ग्रेना के बीच में ये पतला सा एक ब्रिज दिखाई दे रहा है. This is called stromal lamellae. ये इन दोनों को, इन एक ग्रेना को डिफरेंट ग्रेना से कनेक्ट करता है, right? Okay, now we come to the ribosomes. Ribosomes को universal organelle कहा जाता है. Why? Because it is present in all the organelles, right? So these are tiny organelles without a membrane. इसके ऊपर कोई membrane नहीं है. छोटे organelles हैं और ऐसे they are scattered within the cytoplasm. These are the site of protein synthesis. एक ये फ्री स्कैटर्ड इन द साइटोप्लाज्म प्रेजेंट होते हैं. दूसरा, इफ यू जस्ट रिकॉल, हमने प्रीवियस वीडियो में देखा था कि ये किसी के ऊपर प्रेजेंट होते हैं. किसके ऊपर प्रेजेंट होते हैं? ER, yes, एंडोप्लाज्मिक रेटिकुलम. कौन सा एंडोप्लाज्मिक रेटिकुलम? RER, because it has got the different surfaces become rough because of the presence of these ribosomes, right? So they are present in two manners. One, it is attached to the RER. The second, it is lying freely in the cytoplasm. It composed of RNA and protein, and it is the site of protein synthesis, right? Now, sedimentation coefficient. अब इसके साथ हमने पढ़ा है कि S एक यूज किया जाता है, which tells about the. So the ribosomes, they are very tiny particles which are present in the cytoplasm and on the surface of RER. So they are measured in this form, which is called as the sedimentation coefficient, right? So it is denoted by S, that is Swedberg's unit, which is named after the scientist who's worked on it. Now, eukaryotic cell में ribosomes are a little bigger inside and we denote it by ATS. And in the prokaryotic cell, it is 70S. अब ATS का मतलब नहीं, ये नहीं है कि उसका molecular weight है या mass है. It depends on many factors. So you might have studied this in your textbooks also, कि eukaryotic में ATS होता है, which is a composition of 60 and 40, right? So, but it doesn't mean कि वो 60 or 40 compile करके 80 बना रहे हैं और कुछ calculation error है. No, it is not like that, right? So it is the ATS is the overall size of the ribosomes, right? So which is smaller in case of prokaryotic cells. Now, इसका significance क्या है? Significance we've studied earlier also that it is the site of protein synthesis. It provides enzymes also that works on the protein synthesis. As you can see it in the diagram, ये दो units में होता है, the bigger one and the smaller one. So the larger unit here, इसको 80S अगर बोला तो ये इसको अगर 60S बोला तो this one is the 40S. So likewise in prokaryotes it is 50 and